All right. Looks like we've got a full house here. Hi, everybody. Uh, my name is Jake, and I'll be your host for today's virtual event on strain improvement. We are here today because you have a product that you make or that you want to make with a strain. Could be any strain, bacteria, yeast, fungi, algae, mammalian cell lines. Maybe you already have a strain in mind. Maybe you want us to find it for you. But whatever it is, you see an opportunity that depends on the performance of the strain. You want it to grow faster or eat a cheaper feedstock. You want it to thrive in a particular environment, to make more product or to make less waste. So my job today is to show off Ginkgo's tech stack for getting the strain you need to hit those performance targets. And in particular, we're going to focus on two pieces of tech that use evolution or selection to deliver that enhanced performance. This is a little unusual for us because Ginkgo is the synthetic biology company, so we love doing the cell programming directly. Usually we want to be in there writing and editing the DNA. These technologies, they don't require that. We can do evolution and selection together with strain engineering, but we don't have to. So if your product is in a place where certain kinds of genetic modification are not allowed, this can be a regulatory friendly option. It could be a non-GM option for you, and Ginkgo can be your partner for that. So let's talk about Ginkgo. We are over 1,200 people. We're based in Boston. We have offices in California, France, Netherlands, Switzerland. If you come by and visit our headquarters, you'll see a lot of robotics and a lot of automation. That's our foundry platform. Between the technology investments and the physical scale, we've invested almost $500 million in building out this foundry. And we didn't do it so that we could develop our own therapeutics or our own agricultural products or our own chemicals. We did it for you so that we can make the strains that you use to make those products. We are capitalized to scale, and that makes now a good time to partner with us. A good way to think about our business model is we're a CRO. You can send us your R&D projects on a contract basis. So if you need more, you spend more, and if you need less, you spend less. But we want to be more than a traditional CRO in the sense that you can come to us and get things that you can't get anywhere else. Okay, so part of that is scale. We're just bigger than the competition. We can throw the power of the foundry behind your project. At the foundry scale, you can just do things that a smaller lab can't, and our partners get access to that. Part of it is scope. So we want to be an end-to-end service provider. You can come to us with nothing but an idea, and we can execute the entire R&D process all the way through to the technology transfer to your preferred manufacturer. Or if you need less than that, we can do less. But what you don't want to do is farm out little pieces of work here and there to a bunch of isolated teams. Biology doesn't want to be done that way. Biology wants to happen in a foundry. At Ginkgo, we can do all of your strain stuff so that you can focus on your product. The last thing that Ginkgo brings to the table here is the tech. We have an in-house tech stack that you just can't get anywhere else, and that's what we're going to show off today. All right, let's get to the agenda. First of all, the offering. What can we do for you? I just made that pitch. Hopefully you liked it. Then the technology. Today we're going to do a deep dive on two strain improvement technologies. One is called adaptive laboratory evolution, or ALE. The other one is encapsulation and screening, or NCaps. Markets. Who do we work with? Well, obviously we work with you. We work in all the industries that biology touches, but we'll name some specific markets that we think are going to want this tech. I'll introduce you to the BD reps who cover those markets for us. Then case studies. So this is new tech, but there's a track record here, and we'll show you some examples of how it's performed in 3 real projects. That's real data, real outcomes, real customers, and all that good stuff. And then finally, Q&A. We're here to answer your questions. And so for that, I want to call your attention to the Q&A option at the bottom of your Zoom screen, maybe over here. you can type us a question anytime. We've got a big team here to answer them. So happy to have questions about business, tech, or anything on your mind about working with Ginkgo. All right, let's get to the good stuff. The technology. The encapsulation and screening tech that I'll describe came to us through FGen. That is a Swiss company that we acquired in March of 2022. And the adaptive laboratory evolution tech comes from Altar. That's a French company that we acquired in October of 2022. So this event is very timely in the sense that these are two newly acquired technologies for Ginkgo. First up, we have ALE, adaptive laboratory evolution. In ALE, we want to improve the growth rate of a particular strain under particular media conditions. And we're going to do it like nature would, using evolution. The concept is very simple. We're going to maintain a cell population in continuous culture with media flowing in and media flowing out. The cells that grow poorly are washed away. The cells that grow well take over the population. And then gradually, we increase the selective pressure. We make the conditions more similar to what the strain will face in your commercial process. So evolution works. Evolution works. I don't need to explain that for this audience. That's not the technology here. The technology is in the automation. Anybody who's worked in a biology lab knows what a huge pain in the ass it is to maintain a strain culture for an extended period of time. You have contamination, you have washout, you have biofilms, and just the technical error that it seemed small over one cycle, it adds up over 100 cycles and it kills you. Okay. Automation fixes this and it speeds it up. With automation, you can keep the bacteria perfectly balanced between growth and death, where evolution is the fastest. The ALE tech cracks this automation. It took years of engineering to get it right, and that's why I can only show you this tiny little image of the machine. This is automation. You can't get it anywhere else. Okay, so why do we like ALE? First of all, works with many strains. Anything that we can grow, we can evolve. We especially like this for bacteria, yeast, and microalgae. Requires very little prior knowledge. So if you're working on an unusual microbe, for example, you might not have access to very many rational engineering tools, but anything that lives can evolve. It works with natural variation. If your strain needs to be non-GM for regulatory reasons, it works with that. Automation works, evolution works. There's a solid track record behind this tech. What are the limitations of ALE? First limitation here is that we need to select on growth. The growth of your strain on your media conditions. We're limited in our capacity to optimize for other traits. We can still get clever. Maybe we delete some genes, maybe we add some genes. We can use tricks like an anti-metabolite selection to push the evolution in a particular direction. But at the end of the day, evolution is about survival and replication. So this tech is going to deliver cells that survive and replicate better than what you started with. Now let's talk about NCaps, encapsulation and screening. If ALE is about the power of evolution, NCaps is about performing artificial selection at scale. We start with a culture of microbes, just a regular culture of microbes. We want to introduce some diversity. So this could be a rationally engineered library, could be more traditional chemical mutagenesis. We can get fancy, but we don't need to because it's a non-GM friendly approach. The NCaps system is going to encapsulate those cultures at a single cell level into tiny little microfluidic droplets. They call them NLR for nanoliter scale reactors. NLRs can be cultured, allowing the cells to grow and metabolize and make their products. We sort the cells using fluorescence, and we collect the top performers for genotyping and for downstream analysis. I love the NCaps technology. I think it's so elegant. I think that working down at the microfluidic scale is the future. First of all, it's just mind-blowing how many of these droplets we can handle and how quickly. But you get this great consistency. So the engineering steps that are messy at the scale of human pipetting, down here it's the physics that's really in control. Okay, I'm gonna show them off here now. Look at them. Look at them. Look at these beautiful little nanoliter scale bioreactors. Look at them. Oh, I like it. Headline number, 1 million. That's how many of these we can screen in a single run. We can encapsulate almost anything, many different cell types, even mixed populations. We're working down at the single cell level, but it's not a flow cytometer. Okay, so the NCaps droplets, they're more like little bioreactors. The single cells can grow into microcolonies, gives a stronger signal. You get less of that single cell noise. And the other beautiful thing about these NCaps droplets is that you can assay the extracellular environment. Imagine, for example, you're looking for a secreted product. Okay? You can't get that in a fax machine. What are the limitations? What are the limitations of NCaps? The main one is the assay. So we have to convert whatever we're looking for into a fluorescence signal. Although the good news there is that fluorescence is already the signal for a lot of biological assays. It's pretty powerful. You can do a lot with a fluorescence signal. The other one is just the physics of the droplets. So we have to work in conditions where the droplets are stable. So if your conditions are particularly harsh, like detergents or high temperatures, maybe it doesn't work in NCaps. Okay, so we've done selection, we've done evolution. Now what I want to do is zoom out for a second and put things into context. What are these good for? What are the biological products that you can make now that you couldn't before? So I know there's a lot of smart people on the call. I want to get those creative juices flowing. And so for that, I need a little help. I'm calling on the BD team. These are the people who know how to connect these technologies to the needs of a particular industry. So if you're coming to us from agriculture, from industrial biotechnology, from nutrition and wellness, or from biopharmaceuticals, these are the folks who speak your language. You can contact them directly, or you can hit us up strains at ginkgobioworks.com, and we will connect you to the right person. First up, we're going to be talking to Brennan. Brennan Duty has a BA from Ohio State University and an MBA from Cornell University. He has 15 years of experience in sustainability and agriculture and precision fermentation at companies like Land O'Lakes and Cargill. He's also a founder. He started his own company using enzymes to control pests in agriculture. Brennan, welcome.
Thanks, Jake. Good to be with you today.
All right. I will remove my spotlight. Let's see. There we are. Beautiful. All right. Tell me what's going on with ag at Ginkgo. So I know we've seen a lot of growth in that area lately. What's the ag news out of Ginkgo?
Oh, yeah, we have. Absolutely. So last year was a marquee year, if you will, for Ginkgo in terms of agriculture. We made 6 acquisitions. Part of that was the NCaps team, or Encapsulation from Switzerland. But probably the one that most folks are familiar with on this call is around the acquisition that we made for Bayer's Biologicals R&D division. So what this means is that before we, of course, could do discovery, optimization, engineering, and fermentation, but we all did this from as a synthetic biology lens only. And now with this acquisition, we have plant scientists, greenhouse capabilities, and have a really thorough understanding of what it means to do the development end-to-end of a product in agriculture and biologicals, and applying biology to do that. And by taking advantage of those facilities out in West Sacramento.
Beautiful, beautiful. Tell me more about tell me more about these biologicals. So what can biologicals do, and how are they going to solve challenges that farmers are facing?
Yeah, sure. So before we get into sort of the challenges that farmers are facing, let's have Ginkgo define what we mean by biologicals. So biologicals for Ginkgo are compounds that are found in nature that are already produced by or could be produced by microbes. So that means anything that's isolated from the environment that excludes, for example, alginates, which are very common in agriculture. Why biologicals are interesting in this case is that synthetic chemistries are really running up against a lack of research and development in the past 20 years. And the universe of what microbes can actually provide, given that they've been co-evolving with plants in the space for millions of years, means that the opportunities are endless for microbes and these secreted compounds to solve biological problems like pests and disease resistance. And so why biologicals have a really promising future is that, one, we can help overcome resistance. So there are some synthetic chemistries which are overcoming or facing resistance, excuse me, as it relates to certain pests. And two, we also see a lot of consumer and regulatory pressure around the globe for more safer and efficacious solutions that could help support sustainable agriculture, and biologicals are really the solution to that. So when we talk about biologicals with our clients, a lot of them think about them in 4 generations. So the first generation is a microbe that is already secreting this sort of this metabolite of interest. Maybe it's a plant growth hormone, maybe it's a fungicide. A second generation is really what we're talking about today, which is a mutagenized or a lab evolution type microbe, which can secrete yet more of the metabolite. The third generation is cisgenic engineering, and the fourth generation is transgenic engineering. So those are the 4 different types of generations that we're thinking about when we think about strain optimization within biologicals, and that's where Ginkgo is working with biologicals and strain optimization.
Brandon, what makes a good technology offering in ag? What is a customer in ag looking for when it comes to a successful strain improvement project?
Yeah, so there's lots of different ways to contemplate that, but I think for today, the area that we find the most interest in is really around that Gen 2 concept, right? So it's something that is enjoying a really quick to market way, and that couldn't be done before. So when we're talking about NCaps or ALE, we're talking about a strain that can be optimized in less than a year. So really good time to market. The second thing too is that when we do this sort of optimization through mutagenesis and fermentation, we can get anywhere from 2 to 20x, and these numbers that we've seen in our West facility as well as others, for more of a secretion of metabolite. And then third, because we're talking about evolution or mutagenesis, these things enjoy regulatory approval virtually worldwide. So companies can deal with one SKU as opposed to multiple SKUs across different geographies. And that's really the value proposition, to be able to deliver something that's far more potent in a time period that matters and in a way that's going to enjoy regulatory approval worldwide.
Brandon, thank you so much. That sounds great. Now we're going to turn to industrial biotech. And on that topic, we'll hear from Jesse Dill. Jesse is a protein biophysicist. He's got a PhD from UC Berkeley in protein folding and an MBA from UMass Amherst. He's worked at Bolt Threads, at Beyond Meat, at DARPA in the Biological Technologies Office, and then at North Pond Ventures before coming over to Ginkgo. Jesse, let's talk industrial biotech.
Yeah, absolutely. It's an exciting time. In terms of what's driving the industry right now, it's a lot of different pressures. Companies are asking and customers are asking, how can we do things in a more circular and sustainable fashion? How can we do more stuff with industrial waste streams? Companies want to capture as much value as possible, and if you can capture value by reprocessing a waste stream and converting it to something valuable again, then that's very attractive to companies for the bottom line. But customers also want to see that as well. And so finding ways to lower costs of production for existing processes, but also create value of previous sort of wasteful processes, are all on the table right now, and companies are exploring more and more biological ways for doing that.
Yeah. So when I think about industrial biotech, I think cost first, right? Like nobody likes cost, but industrial biotech particularly looking to lower those costs. And I know one way to do that is with scale. So tell me a little bit about Ginkgo's scale-up capabilities. If I like these improved strains that Ginkgo delivers, can I get them 10,000 liters at a time?
Yeah, absolutely. And at Ginkgo, our philosophy is we want to start the project where you need to start it and carry it as far along that process, taking out as much technical risk as we can along the way. And so if that means improving something out at the bench scale and then going to bench scale fermentation or to pilot scale, we have the facilities for that in-house. If you need to go to higher volumes though, and you want a process that's fully vetted at 10,000 or 50,000 liter volumes, we've done that in the past as well, and are happy to partner with you and outside contract manufacturers to hand you a robust process and a robust organism that are ready to hit the road. Cost, as you noted, is everything. And, sometimes customers come to us with an existing process where we can actually drop in these methods and tune up that organism a little bit further, make it a little bit more fit, or improve its productivity even farther than has already been done. And, we're exploring the right sort of deal structures and technical scopes of work that solve those needs and help customers get more out of an existing process. Or if we're starting from something earlier, carry it all the way up to the volume that's needed.
I love it. Thank you so much. Thank you so much, Jesse. Now we pivot to nutrition and wellness space. We're going to hear from Sid. Sid Kishore is a biologist with a PhD in cell and molecular biology from the University of Pennsylvania. Before Ginkgo, Sid worked in strategy consulting at Bain & Company. He's passionate about nutrition, wellness, and food sustainability. Welcome, Sid.
Thanks, Jake.
All right. Tell me about food. Tell me about nutrition and wellness. What kinds of innovation can synthetic biology bring to nutrition and wellness?
Yeah, that's a great question. So biology in sort of food and nutrition is nothing new, right? We've been using biology in this field for millennia now. Think about brewing beer, baking bread, and even over the last century, the discovery of vitamins that have really spurred the growth of human health. What's really interesting over the last sort of few decades is that we understand biology a little bit better, and we have developed tools to engineer organisms to really leverage biology for our applications. So in the field of nutrition and wellness, this is being used right now for production of certain proteins like dairy proteins or egg proteins, functional alternatives to animal sources, for producing flavor and fragrance molecules, for producing fats and lipids more sustainably, for producing probiotic strains to really spur human health and wellness. And these are only going to accelerate in the future, and it's something I'm super excited about.
Mm-hmm. So let's zoom in a bit on specifically the strain improvement tech that we're highlighting today. So how can our strain improvement capabilities help our partners in nutrition and wellness?
Yeah, I see a bunch of different opportunities, and we're working on these currently, but it probably falls into three buckets that I see. So one is improving strains, producing proteins, enzymes, small molecules of interest. Two is improving strains as a product themselves that go into probiotics or microbiome. And three is something we could have a conversation for hours about in terms of what's important to the consumer, which is non-GM in this field, right? So these are the areas where I see the tools that we're highlighting today, NCaps and ale, being really, really powerful. So on the first bullet there, oftentimes to improve a strain to produce a protein, let's say, for instance, requires really understanding the complexity of biology. So this, you may have to screen thousands of different strains in parallel, and that's something Ginkgo's platform is really set up to do. It's really hard to do at a bench scale, as you were talking about earlier. You get one colony, and it's probably a bust. Ginkgo does this in a highly parallelized fashion. Similarly with probiotics, ale and NCaps, and I know we'll talk about this with a few case studies later, really, really powerful tools for non-GM improvement of certain strains that go into food products, which is absolutely crucial for consumers that our partners are serving. And that's really one of the strengths of this technology that I'm very, very excited about.
All right. Thanks a lot. Thanks a lot, Sid.
Thank you.
Now, turning from nutrition and wellness, last but not least, we have biopharma. So we'll be hearing from Kaylee Carpenter. Kaylee has a chemical engineering degree from the University of Wisconsin and an MBA from Harvard. She's worked across the healthcare space for 8 years, notably at AbbVie, before coming to Ginkgo. Welcome, Kaylee.
Thanks, Jake. Excited to be here.
All right, so pharma, particularly large sector in biotech. Can you break it down for me? Can we zoom in on some potential use cases for better strains in biopharma?
For sure. A lot of Ginkgo's abilities to make strains to produce and transform things, molecules in both microbial and mammalian systems are really relevant in the pharma space. For example, our strains are used by process development and CMC teams at places like Merck to make APIs using enzymes for biocatalysis. And they're also used to biosynthesize natural products where sustainability, supply chain, et cetera, issues can be of concern. We're doing that in our partnership with a company called Saponics, where we're sustainably producing QS21 adjuvant.
Ah, okay. I heard a lot in there. But I heard mammalian. I heard keyword mammalian systems. And so I know when I think about biopharma, I'm definitely thinking a lot about mammalian cell lines. So can you expand on how our capabilities are relevant to mammalian systems?
Yes, yeah, and apologies if my video is frozen. Yeah, we're particularly excited about the possibility to combine discovery with manufacturing using the NCaps tech. This technology essentially allows innovators like you all to sort of skip a step or two in your biologics development. We essentially can define interesting libraries of antibodies, produce them in a cell line, show, for example, and screen them for activity all within the NCaps setup. And we can also do this, all this antibody expression and screening in manufacturing relevant conditions, and then only actually select for cells that are up to the challenge of high expression in these complex media or process conditions. And then, as we've said, these techniques can also be used in microbial systems too, as was mentioned earlier.
Okay, I like that. So it's kind of a neat trick, right? So you think, well, this is an antibody engineering project, but we can turn it into a cell screening project by expressing those antibodies in like a CHO cell line, for example, and then screening them with the NCaps system. So I think that's really fun. I think that's a way that people kind of hack this technology, use it in ways that you don't necessarily expect.
Totally.
All right. So now, okay. So what about live therapeutics? What about when the strain itself is the drug? What about when the bug is the drug? What kind of strain improvements do we need for a living therapeutic?
Yeah, totally. And I know there's a question on microbiome in the chat. Like we've said, these technologies can for sure be used to make strains that make both large and small molecules. But you're totally right. Sometimes the strain itself is the product, as in Synlogic's case, for example, one of our partners. And there's a lot we can do when the bug is the drug. We can first engineer the strain to do or make something in the body, like we've said. We can also essentially give the strain superpowers, whether it's an engineered strain or a wild type using the ALE, Adaptive Laboratory Evolution tech. We can also evolve strains to at least retain and in some cases enhance activity in unfriendly growth conditions using ALE. So extremely low pH, for example, which is often relevant in the microbiome context. And we can also run a mutagenesis campaign on a starting strain and use the NCaps tags to select only the variants which produce high quantities of the target product in, again, the intended final processor in vivo conditions. So there are lots of ways to pair up these technologies too, both with and without strain engineering for live biotherapeutics, consortia products included, in addition to all the mammalian cell use cases we've discussed already.
Now we've heard a little about the tech. We're talking about markets where it can enable new products or improve your bottom line. Next, we're going to present 3 different case studies that show how the strain improvement works in practice. We've got one for ALE, one for NCaps, and one that combines them with Ginkgo's rational design capabilities. And so for this segment, we're going to turn from our business experts toward our technical experts. These are the folks who are really in the foundry delivering these strain improvements. First up, we welcome Sandrine. Sandrine Gosling has a PhD in organic chemistry from Orléans University in France. She worked doing bioengineering for the French Atomic Energy Commission of Saclay before joining Altar in 2018. Now she works at Ginkgo on the implementation of ALE projects as a senior engineer. Sandrine, thank you for joining us.
Thank you, Jake. This case study features our work with Braskem, which is a leading chemical company in Brazil that makes plastic polymers. The starting point for many of these plastics is monoethylene glycol, or MEG, which Braskem wanted to produce using biotechnology. Braskem also produces ethanol from sugarcane, and this process generates a lot of bagasse, the lignocellulosic material of plants that is still rich in sugar, like xylose, but can be easily or economically turned into ethanol. Although MEG can be easily sourced from petrochemicals, finding biological pathways to produce MEG from sugar sources like glucose and xylose in bagasse can, one, reduce waste, and two, also reduce dependence on petrochemicals, both meet their sustainability goals. So Braskem's internal R&D team had engineered an E. coli strain to produce MEG from glucose and xylose from bagasse. Without getting too deep into the weeds, wild-type E. coli has evolved to grow on glucose, and convincing it to prefer a different sugar is very challenging. So next slide, please. Braskem's engineered E. coli strain was capable of growing off of xylose and glucose simultaneously, but the strain had a delay in xylose uptake and consumed xylose at a lower rate than glucose. And this is where we deployed adaptive laboratory evolution, or ALE, to approach this industrial challenge. Our team tried two evolutionary approaches to improve upon two phenotypes. The first, reduce the time delay. That is to say, evolve the strain to consume xylose immediately, even with glucose present in the medium, and increase the rate of xylose consumption so that glucose and xylose are consumed at comparable rates. The ALE approaches yielded significant changes. We were able to successfully evolve a strain that starts consuming xylose and glucose simultaneously at the start of the fermentation batch. Then the average uptake rate of xylose significantly improved. By the time all of the glucose was depleted, 96% of the xylose was also depleted. And previously, only 64% of the xylose was consumed. And so this is a 50% improvement. And the final strain was also more efficient at consuming both glucose and xylose, and batch duration was decreased from 23 hours to 7 hours. For those who are listening in today, with any application in mind, adaptive laboratory evolution can deliver value in many different ways. First, it's a powerful strain improvement tool when rational engineering approaches have reached their limits or you're hitting roadblocks in your project. Then it can deliver significantly improved phenotypes in fairly short periods of time. Indeed, we started to see a competitive strain in 5 months for one of the ALE strategies we tried. And finally, it's an approach that doesn't require targeted gene editing, which for many industries can confer fast regulatory acceptance. So if you have more questions about this technology, I'll be available to answer a question in the Q&A.
Thanks so much. Thanks so much, Sandrine. So for me, this case study highlights these really drastic improvements that we can sometimes get with adaptive laboratory evolution. I think that most of us know that the strains that we use are not perfectly adapted to their environment, but it's like, maybe they're close, right? They should be close at least, but it's not always the case. Sometimes evolution can really just knock your socks off. Next up, we'll be hearing from Rocio Aguilar Suarez. Received her PhD from the University of Groningen in the Netherlands. She was a researcher in the microbial proteomics group and the Institute for Systems Biotechnology, both in Germany. Rocio joined FGen in Basel as a research scientist on the topic of data-driven cell factory engineering. Now at Ginkgo, she's a research engineer specialized in strain engineering and ultra-high throughput screening. Rocio, thank you so much for joining us.
Thank you, Jake, for the nice introduction. So this collaboration started with our partner that had a new peptide ingredient that they wanted to commercialize for human nutrition. They did a lot of rational design-based engineering on their pick strains in-house to get high levels of production, but there is always a but, they couldn't get the improvements they needed to lower costs. That is the moment when they approached us to try unbiased strain development. And to solve this challenge, we combine random mutagenesis, in this case, both chemical and physical, and ultra high throughput screening, encapsulation and screening, or strongest part, to deliver a strain that produces 50% more of this valuable peptide. Next slide, please. So this is how we did it using the NCaps technology. Before diving into any screening, we first need to develop an assay that could reliably measure protein secretion in the nanoliter reactors that Jake described before. So I am talking about what you see at 6 o'clock in this diagram. Once that we have an assay and it's already set, we generated a library using random mutagenesis of only 200,000 variants and selected the top performers among that population. Those ones, we took them and test them for other characteristics and choose the best one. And starting from this one, we did another round of mutagenesis, so again the cycle, and we, in this case, screened only 290,000 strains and picked again the top hits. We screen thus also for secondary characteristics and choose again the best one that we use for a final third round of mutagenesis, this time testing 260,000 variants. We send to our customer the best 4 candidates and they try them in their own bioreactors. They confirmed the 50% improvement in their titer among those top candidates. The screening part of this project took approximately 4 months, and in total we spent like 10 months to deliver an enhanced strain using non-GMO strain, improvement techniques, and our encapsulation and screening system.
Rocio, thank you very much. Thank you very much. I appreciate the flex there of, oh, we only screened 260,000 variants in the second round. I appreciate it. I have a couple of quick questions before I let you go. First question, what organisms does this work with? Can we really encapsulate anything?
Well, you see with the variety of microorganisms that we work, so I just want to make sure that this screening technology can work with just about any host organisms. I'm talking about including mammalian cells and filamentous fungi. So this is pretty cool because the ones that work with filamentous fungi, they know that they are not so easy to handle, and we can work with them.
Very cool, very cool. All right, thank you. Thank you very much, Rocio. For the last case study of this segment, we turn to Rose Berthomme. Rose has a master's degree in synthetic biology from the University of Évry near Paris, specializing in metabolic engineering. She's been at Ginkgo for 4 years, working on strains that produce small molecules as well as live cell probiotics. Rose, thank you so much for being here.
Thank you, Jake. So our partners in Logic developed living medicine. They basically engineer probiotic bacteria to be ingested so they can act in the gut of humans. And they had been developing a strain to treat homocystinuria, or HCU, which is a rare disease in which humans cannot digest methionine properly. And this disease leads to high level of homocysteine in the blood, which cause various symptoms such as ocular or neurodegenerative disorder, osteoporosis, or intellectual disability. And currently, the only way to treat HCU is through restrictive diets and vitamin B6 or B12 supplement. So Synlogic was developing an E. coli nasal strain that could metabolize methionine in the gut as a more robust treatment. And they came to us to improve the strain so they could take the treatment to phase 1 clinical trials. Next slide, please. So we identified the enzyme that could import methionine into the strain and also metabolize it away. And using our computational tools and our proprietary data from past project we had with Synlogic, we designed metagenomic library for this enzyme. And in parallel, we also developed high-throughput assay to identify the best enzyme in each library. And we wanted to answer 2 questions. Which of these enzymatic methionine importer are the best at importing methionine, and which of these pathway enzyme are the best at metabolizing methionine? So we screened more than 2,600 enzymes to identify the best performing enzyme along the methionine importer and the metabolic pathway. And then we finally redesigned Synlogic E. coli nasal strain with multiple combination of the top performing enzyme, and we screened a combinatorial library of 22,000 strains. And as a result, the best performing strain showed 3 to 4x improvement in methionine consumption as compared to the Synlogic starting strain. This strain has achieved proof of mechanism treatment for HCU and positive result in the phase 1 trial. And right now, the FDA has granted orphan drug designation to the strain for the treatment of HCU. If you have more questions about this case study, you can ask us in the chat or just send us an email. Thank you.
All right. Thank you so much. Thank you so much, Rose. So for me, the HCU case study, it really shows off how Ginkgo's customers benefit from us being end-to-end. We can just do it all. We can do it all. Sometimes rational engineering is right. Sometimes selection or evolution is right. And then when we need to combine them for maximum effect, we have all the technology that we need under one roof. And that's what it takes to win at these really, really demanding pharma projects. So I think it's really exciting to see. All right. And that is going to conclude our case study session. And we're going to move on to the final Q&A session for this event. So one more time, I will point out again the Q&A feature at the bottom of your Zoom. We've seen a lot of good questions coming in. We'll keep answering them. For this segment, I need to bring in some more technical heavy hitters. So we're bringing in the leadership from Altar and from FGen, who were instrumental in the development of the technologies that we're sharing today. We welcome Simon Trankar, co-founder and CEO of Altar, now the head of ALE at Ginkgo. Welcome, Simon.
Hi, thank you, Jake.
And Andy Meyer, co-founder of FGen and head of overall activities at the company, now at Ginkgo as a senior foundry lead. Welcome, Andy.
Hi, Jake.
Let's start with you, Simon. Okay, so some technical questions. What's the typical duration of an ALE experiment? And what are the factors that influence how long it takes to do a laboratory evolution?
Yeah, that's a very good question, and which I've been asked in the Q&A here. Well, it's like if you ask a taxi driver how much time it will take to arrive. I would ask first, okay, where do you want to go, to be able to give a fair estimate. But The best answer I give to this question is the typical duration of ale experiments that we've conducted in the past 10 years, which ranges between 3 and 6 months usually. Sometimes it's more. In academic environment, it can be even longer. The main driver for this is the doubling time of the starting strain, because we will rely on mutations that will occur mostly naturally through when cell divide, and of course over a given period of time there will be more generations if the bugs grows faster, right? So, but yes, I think the right answer here is 3 to 6 months most of the case.
All right, I know where we're going. I know where we're going in this taxi. We're going to Strain Improvement City. Am I right?
Yeah, I think it's good also that at Ginkgo you have all the analytic tools that would be like the right GPS, right, to the taxi driver, so that I think that you can always collect samples and test them and assess the performance of the strain against the target and evaluate whether this can be interrupted or still to be continued.
Mm-hmm. Okay, another question. What can you select for? So what are some features of a strain that you might want to enhance using ALE?
Yeah, so I think that what's important to remind is that the core mechanism for selection is continuous cultivation. So we at constant volume. So we had sterile medium to the cultivation chamber, and we keep that volume constant so that we have to remove suspensions, right? So there is a trade-off between growth of the population through cell divisions and dilution of this population through the withdrawing of a fraction of the suspension. So it's a Darwinian competition to remain in place in the system, where only the bugs that can thrive at a given pace will actually have a higher probability to survive. So what we can select for is our traits coupled to growth or survival. And so obviously better utilization of substrates, use of alternative substrates, tolerance to toxic compounds, low pH or high pH or higher temperature, adapt the strain to different cultivation conditions is definitively doable for any program. And then sometimes we might be able to engineer some tricky ways to tie growth to production. And in many cases, the yields are limited by product tolerance. So by enhancing product tolerance, you actually are targeting higher yields. So that's typical applications for ale.
Great, thank you so much, Simon. Thank you so much. Now turning to you, Andy. Turning to you. Well, I've got a good one. For Andy. All right, so let's talk about screening. Okay, we're talking about screening technology. All right, here's my question for you, Andy. Haven't you heard the news? Machine learning is going to solve biology. We don't need data anymore. We don't need data. Why do we need high throughput screening in the age of machine learning guided biological design?
For many reasons, of course. So on one hand, I don't see it as a opposite technology. I see it as a complementing technology. So with AI, we can really determine a lot of targets that we can engineer. And by using combinatorial design, we can make large libraries, and then we end up with high throughput screening. So that's a very obvious reason. On the other hand, I don't think that AI is really out of its infancy for non-model organisms. So it's really working fine if you are on the standard organisms like Cerevisia or E. coli. But if you go to non-standard organism, it gets a little bit more tricky. So there, I think high throughput screening is needed. And last but not least, also if the regulatory components comes into play. So if your product has to be non-GMO or you don't have engineering tools for the organism to modify, then of course you end up with large numbers of clones that you want to analyze. And therefore, I think even in the age of machine learning and computational biology, which we also have nice tools at Ginkgo, I think high throughput screening will still be used for quite some time.
Okay, that makes sense. You convinced me. You've convinced me. We still have jobs.
I can keep my job.
Another question: when we do the screening at a nanoliter scale, how does that transfer to the kiloliter scale? What can we do to ensure that the screen that we're doing in these nanoliter scale reactors is screening for the actual phenotypes that we need when we're gonna scale up these strains for production?
Yeah, I mean, scale up. Everybody on this call probably knows that scale up is an issue. Okay, so what we can try to perform our screenings as close as to the application conditions as possible, and I'm absolutely convinced that our platform, so the nanoliter reactors, is one of the technologies that are closest to the final applications conditions. So, for example, we can put our nanoliter reactors in a little bioreactor during screening. So already are one step closer to the final bioprocess than, for example, if you do the same procedure in microtiter plates. So I think screening as close to the application is key to mitigate the risk of the scale-up.
Thank you. Awesome. Thank you so much. Thank you so much, Andy. I have a lot more technical questions. I know that there's a lot more happening in the chat function. Unfortunately, we're not going to be able to get to everything live, but I know we have people typing away to answer those technical questions in the chat. And so with that, I will say the time has come to wrap up this virtual event. The time went by fast. Some highlights from today's event. Today was a spotlight on two technologies that we use to do strain improvement here at Ginkgo, one using evolution, one using screening. These technologies are non-GM if that's important for your application. They work at the whole organism level. They use natural variation. Nevertheless, they can be very powerful thanks to this enabling layer of automation. Continuous evolution over 100 generations, encapsulation of 1 million variants at once. Okay. So if you like what you see today, if you want better strains, these are the people who can get your project on the Ginkgo Foundry. Brennan, Kaylee, Jesse, Sid, operators are standing by. Put that $500 million worth of Ginkgo Foundry infrastructure to work on your strain. You can email us strains@ginkgobioworks.com. Strains@ginkgobioworks.com, and look at this. Look what I got. I just got this working. Here we are. Strains@ginkgobio, right? There's your call to action. Strains@ginkgobioworks.com. Thanks again to everybody who asked a question in the Q&A. I've got one more personal favor to ask. After this Zoom call ends, you should get a link to a survey, 6 multiple choice questions, collect a little feedback on how these virtual events are working for you. If the feedback is good, we'll do a lot more of these, and hopefully we can, because I'm having a blast. All right. So until next time, as we like to say around here at Ginkgo, let's grow. Looks like we're at 100, 130. While we're waiting, I'll share a quick story about strain improvement that it's a project that I did back in grad school. So I was studying hydrogenases. These are the enzymes that metabolize hydrogen. And I was trying to engineer a strain that could consume hydrogen as an energy source. So I set up a selection to improve my strain, right? And so in those days, you had to do everything by hand, right? I was going to plate out a bunch of bacteria under an atmosphere of hydrogen and oxygen, and then see what grows. So the problem with this, of course, is that atmosphere is also explosive. So I got these little vacuum chambers that you can put the Petri dishes in, and I add a bit of hydrogen from a tank, And then I let them sit in the incubator to see if anything grows. And after like maybe about 3 weeks, I get one little colony, right? You can kind of look in the chamber and be like, oh, maybe there's one little colony growing in there, right? So I pop open the vacuum chamber, and then suddenly everything gets really quiet. And then my ears are ringing because the hydrogen had exploded, killed whatever colonies were in there. And then they didn't let me do that experiment again. They didn't let me do it again. Okay. So, okay, just to be clear, Ginkgo Bioworks, not a part of that story. That was before Ginkgo existed. That would never happen. Ginkgo Bioworks, that is not a Ginkgo story.